Hello YouTube, Joseph here with Prep for STEP. In this particular lecture, we're going to talk about major histocompatibility complex, or MHCs. It's a pretty complicated subject, so hopefully by the end of this series, you will have a great understanding of what MHCs are. Let's try to get through it as soon as possible. Okay, so MHCs, what are they? They are histocompatibility complexes, basically tissue, histo meaning tissue, compatibility is compatibility. So if we were to get trying to get a tissue donated from somebody or a kidney donated for somebody, the donors MHCs would have to match ours, okay? So what are MHCs in the first place? Well, they're cell surface proteins, okay? They're made inside cells. Every single cell in our body, every single nucleated cell in our body will have an MHC on its surface, okay? And what that MHC will be doing is it's trying to communicate with our immune system by presenting peptides that are made inside that cell to the immune system, telling the immune system that, look, I'm a self cell, I belong to this body, please do not kill me and please do not attack me, okay? So this basically is what of the MHCs are going to be doing. They're encoded by an HLA gene. HLA stands for human leukocyte antigen, and as a matter of fact, in humans we do not use the word MHC really, we use HLAs, okay? So human leukocyte antigen. That's a gene on chromosome 6 in humans, and all of these MHCs are presented in a codominant pattern of inheritance, meaning that on each cell surface you will have two different types of MHCs, one coming from the father and one coming from the mother. It's codominant. Neither parent is actually dominant in this particular aspect, okay? Function, what they actually do, they mediate interaction between different immune cells. Like antigen presenting cells can connect or can talk to the other immune cells, or the T cells, in the case of MHC class 2. In the case of regular body cells, the MHC class 1 helps it communicate with the immune cells, telling the immune cells that, look, we're self cells, do not kill us, and etc. There are two different types. In this video, we're going to talk about class 1, and in the subsequent video, we're going to talk about class 2. So let's get started. Okay, so now we're going to talk about the MHC type 1. First off, this MHC is presented on all nucleated cells, okay? All nucleated cells. What does that mean? It means that pretty much every single cell in our body is going to have an MHC type 1 on its surface. It's made in that cell, in the rough endoplasmic reticulum, then it travels to the surface. And why does it do that? Why do we have an MHC class 1 on our surface? Well, it's in order to tell our immune system, or tell our T cells that, look, I'm a self cell, do not kill me, okay? One of the reasons that we get a reaction to donor cells is because they have different types of MHCs. So when the T cell or the cytotoxic T cell comes by, it's actually going to recognize it as a different type of cell and it's going to attack it. So an MHC type 1 is basically telling all of our immune system that, look, we are self cells, do not kill us. So that can be made in a better graph if I draw this out. So let's say this is any type of cell except for red blood cells, right? This is any type of cell. We're going to have an MHC class 1 on its surface. Okay, there's 3 domains, and we'll talk about the structure in a little bit. And then we have a T cell. Now this MHC class 1, once it's made in the cell, it's going to have to travel somehow to the surface, and it's going to present self proteins or self peptides here on this little groove that it's going to present it to the T cells. This is our cytotoxic T cell. We know that cytotoxic T cell is CD8 positive. Now, what is CD8? Cluster of differentiation 8. It's basically cell surface molecules, allows us to recognize what type of cell we're dealing with, because we can't really look at these cells, right? So we have to find a marker for these particular type of cells, which CD8s happen to be a marker for cytotoxic T cell. On the other hand, CD8 does help in the process. So a cytotoxic T cell is just wandering by. It comes and it sees this particular type of cell. It uses its TCR. If I make this, TCR is basically a TCR, or T cell receptor. This is our MHC class 1. And then we have a CD8, which helps these two bind together. This will be our CD8. Okay, so the cytotoxic T cell comes by, looks at this, looks at these proteins. Oh my God, these are self proteins, so it doesn't kill it. If this was, let's say, a donor cell, then this donor cell will have a different type of MHC. It would be presenting different peptides here, and the cytotoxic T cell would basically attack it. But in this case, that doesn't happen, okay? Another scenario would be, let's say this cell was virally infected. if this cell by any chance was virally infected, these proteins that you see presented on the MHC would be viral proteins. If these viral proteins are presented here, then what would you see is that the cytotoxic T cell wandering by with the help of CD8 will bind to this, with the help of TCR and CD8 would bind to this MHC type 1, and it would realize that this cell is virally infected, and then it would attack it again. Okay, so in two cases, this is basically immunology telling that the cytotoxic T cells help protect us against virally infected cells, which is one of the mechanisms of that, and it also helps us against foreign cells. Okay, so basically, again, one last time, MHC class 1 will be made in all nucleated cells, travels to the surface. While it's traveling to the surface, it starts putting some self-peptides over here in this groove, presenting that to the TCR or the T cells or the cytotoxic T cells, and telling the cell that do not kill me, I'm a self cell, I belong to this body, so do not kill me. Okay, so now that we got that out of the way, we're going to go back to our little thing here. The main thing that you have to realize is about MHC class 1 is the type, the structure of MHC class 1, and how it is actually synthesized and how it gets to the surface. That's the key thing. Okay, first off, you have to know that it's synthesized through an endogenous pathway. We're going to go through that pathway in a second, so don't worry about it. Now, let's look at what the structure of the MHC class 1 looks like. Okay, so now let's talk about the structure of MHC class 1. Okay, so there is two different parts to it. There is a heavy chain. All right, there is a heavy chain and there is a light chain. The heavy chain, as you can see right here, this will be our heavy chain, has 3 extracellular components, and this is the outside of the cell, and this is the cytoplasm right here. Okay, so the outside of the cell, or the extracellular, has 3 different components: A1, A2, and A3, and basically these are basically peptides. So inside would look something like that. They're just a strand of peptides. Okay, it's a protein, and the groove between the first 2 extracellular surfaces is location for peptide presentation. So between alpha 1 and alpha 2 subunits, the extracellular subunits between the first 2, which is these 2 guys, this groove is where the peptides are presented to the cytotoxic T cells. Okay, the second portion of it will be the light chain. The light chain is also called the beta 2 microglobulin, and it helps with the transport of this MHC. We said it's made inside the cell and it has to get to the cell surface. This beta 2 microglobulin portion will actually help this MHC reach the surface of the nucleated cell that we're talking about. Okay, so that's basically it. Now, a lot of questions are out there asking, okay, now if the beta 2 microglobulin, for instance, there was a mutation in it, or something happened to it, what would be the likely consequence? And the consequence is that the MHC will not be able to get to the surface. I've seen these in questions before. Okay, so now we're going to talk about the next portion. We're going to talk about the endogenous pathway of how this actually gets to the cell surface. Okay, so in this section, we're going to talk about the endogenous pathway. Okay, we're going to talk about the way that MHC type 1 is actually loaded with those self-peptides. Okay, so first off, we have to know how this actually happens, and it helps to know that it uses a complex called the TAP complex. Now, the TAP complex is used in the endogenous pathway of MHC type 1 loading. It stands for transporter associated with antigen presenting or processing. Okay, so that's what TAP stands for. The TAP is actually located right here. It's just a transporter protein that is located in the rough ER, okay, in the little membrane around the rough ER. Okay, so the rough ER basically looks like this. These are our little ribosomes on the endoplasmic reticulum. The MHC, we said, since it's going to travel to the surface, and any protein that's designated for outside the cell or actually designated for the cell surface will be made in the rough ER. Now, this is our MHC class 1 being made here, but these peptides, the self-peptides, the cell peptides have to be somehow put on this before it makes its way to the surface. And how does that happen? Well, all these proteins, we have tons and tons of enzymes and proteins and all kinds of stuff in our cytoplasm of each nucleated cell. These proteins are routinely degraded by what we call the proteasome. okay? And they turn into small little peptides. These peptides, with the help of this TAP complex, go through this, through the membrane of the RER, or the rough endoplasmic reticulum, and they get put right here on top of this particular MHC class 1. Then the MHC class 1, with the help of the beta 2 microglobulin that we spoke of previously, travels and ends up on the cell surface with these self-peptides. Now, keep in mind that, let's say, the proteins that are getting degraded here in the cytoplasm are viral proteins. That could happen, right? If a cell is virally infected, then the proteins, because basically viruses take over the transcription in the cell to make their own progeny. So these viral proteins, okay, these viral proteins will also be degraded, and they could also be presented on MHC type 1, which helps the T cell to recognize that this cell is virally infected, and then the T cell, with the help of all the granzymes and everything, will actually attack this cell if it's virally infected. Same scenario could be for a donor cell, okay? So this is the endogenous pathway of MHC type 1 loading. All right, I hope this video helps. Please like, share, and subscribe. And check us out on Facebook, Prep for Step. All videos will also be posted there. Thank you.